Bioinformatics is a field of science, in which biology, computer science and IT merge into a single discipline. It is the development and implementation of tools that enable efficient access and management of different types of information. By using bioinformatics tools, various types of biological data can be analyzed and interpreted easily and shortly, from extraction of information from the massive biological database. One of the tools in bioinformatics is BLAST, or known as Basic Local Alignment Search Tool. It is the homology and similarity tool, where it measures the degree of similarity between two sequences. It is commonly used to identify the similarities between novel query sequence of unknown structure of function, and database sequences whose structure of function have been clarified. The PDB-ID, 3BEQ protein sequences can be compared against BLAST protein sequence database. The results from BLAST include similar conserved domain, conserved alignment graph and alignment of query and subject detection. The detected conserved domain for both of the A and B chains of 3BEQ is CADD. It can be seen that both of the results for 3BEQ-A and 3BEQ-B are the same. This is because some protein are composed of identical chains, and some are non-identical. Even with identical chain, the relative positioning controls the symmetry of the complex, and can have biochemical implication. Interpro is bioinformatics resource that provides functional analysis of protein sequences. It classifies them into families, and predicts the presence of domain and important sites, also with their function and sequence features. From the PDB-ID of 3BEQ, glycoside hydrolase family of family 34 and C-alidases domain was obtained. Both have no relationship with other family and domain. Glycoside hydrolase contains 85 different families, and glycoside of family 34 contains only one known activity, which is C-alidase or neuraminidase. Meanwhile, C-alidase and neuraminidase hydrolyzes the non-reducing terminal sialic acid linkage in various natural substrates. Next, active site identification of 3BEQ was performed using Catalytic Site Atlas (CSA) database. The identification of binding interactions and the active site allow the recognition and precise positioning of an enzyme's substrate in proximity to the chemically active catalytic residues, and lower the energy of the transition state, which aids catalysis. The active sites of 3BEQ are as follows. Besides, multiple sequence alignment (MSA) is generally the alignment of three or more biological sequences, protein and nucleic acid, of similar length. It was performed using Clustal X, and the alignment of 3BEQ sequence with other sequence are as follows. In conclusion, the use of bioinformatics in biological research helps in advancing the progress of new discovery and also new knowledge, especially on therapeutic field.